The muscle physiology of a cheetah is definitely one to observe in awe. This amazing animal can reach speeds up to 103 kilometers per hour, making it the fastest terrestrial vertebrate. Its muscles in combinations with its skeletal structure allow it to rapidly accelerate and maintain high speeds, but only for a short period of time.
Attaching to bones by tendons, muscles contract, pulling on the skeletal structure, causing movement. In a cheetah, muscles account for 50% of its total body mass, where 15% is allocated to the forelimbs and 19% to the hind limbs. Due to physical properties, fiber type and distribution leads to the amount of force generated by each contraction.
As seen in all skeletal muscles, the conversion of electrical signals to mechanical force occurs through the excitation-contraction coupling. Actin and myosin proteins of the thick and thin filaments interact to form cross bridges. The filaments slide together, causing a shortening of the sarcomere, creating active force. The release of ADP and inorganic phosphate occur as the head of the myosin continues to pull on actin. This continues to cycle as long as there is an acceptable amount of calcium and ATP available. When ATP attaches to the head of myosin, it can no longer attach to actin. The cross bridges break and the muscle relaxes.
Skeletal muscle fibers are identified based on characteristics in terms of their morphology, biochemistry, and physiology. Generally referred to as slow or fast twitch, fibers are categorized as one of three types: type 1, type 2A, type 2B, or X. The distribution and quantity of the fibers are specialized for the mode of locomotion.
Type 1 are slow twitch fibers characterized by their small diameter and increased myoglobin and mitochondrial content. Requiring oxygen, they rely on oxidative phosphorylation for the formation of ATP. Increased oxygen and energy availability make this type of fiber fatigue resistant with a low metabolic cost for maintenance. However, only a small amount of force is generated with each contraction.
Fast twitch fibers generate large amounts of force and can be divided into type 2A and type 2B or X. Type A are intermediate fibers possessing characteristics of both type 1 and type 2. With increased blood flow, myoglobin, and mitochondrial content, they use aerobic oxidative phosphorylation to acquire energy, making them fatigue resistant. Type B or X have a larger diameter and decreased blood flow in comparison. Relying on anaerobic glycolysis increases creatine phosphate, glycogen, and enzyme activity, resulting in a high susceptibility to fatigue.
When compared to a mammal of similar size and shape, such as a greyhound, the cheetah is able to maintain greater maximum speeds. Studies have shown this may be the result of fiber type distribution. The muscular system of a cheetah is strongly composed of increased proportions of fast twitch muscle fibers, specifically type 2X. The increased contraction rate and power output of these fibers increase their sprinting abilities.
Type 1 fibers are observed in almost all muscles of the cheetah, but are seen in greater quantities in the forelimb of the animal. This relates to function, as these muscles are mainly used for deceleration and knocking their prey off balance while hunting. The three different types of fibers are apparent in each muscle except for the triceps brachii caput medial, which has the greatest amount of type 1 and does not contain type 2X fibers. Similarly, the infraspinatus contains a large amount of type 1 fibers, with only 9% type 2X fibers. The biceps brachii and acromial portion of the deltoideus have an increased number of fast twitch fibers, making them an exception.
The hind limbs of cheetahs generate most of the power for high-speed running. For this reason, the majority of fibers observed are type 2X, which produce the greatest amount of active force per contraction. The vastus intermedius, however, contains no type 2X muscle fibers and over 60% type 1 fibers. Reduced quantities of type 1 fibers are observed in other muscles, especially in the psoas major and rectus femoris, which contain less than 5%. The gluteofemoralis, rectus femoris, tensor fasciae latae, and vastus lateralis house the greatest amount of type 2X fibers. These muscles contribute to acceleration and speed maintenance.
Muscles along the body of the cheetah function in the stability and flexibility of the spine. Increased fast twitch fibers are found in the majority of the thoracic muscles, averaging 32% type 2A and 40% type 2X. Each type of fiber is present in each individual muscle with the exception of the pectoralis superficialis and rhomboideus, which contain only type 1 and type 2A fibers. With increased fast twitch fibers, there is a reduction in type 1. The rhomboideus and trapezius cervicus, which function in the stability of the shoulder, contradict this, observing more than 50% slow twitch fibers. Populations in the pectoralis profundus, longissimus lumborum, and latissimus dorsi exceed 70% type 2X, similar to those located in the hind limbs.
As previously stated, characteristics associated with fast twitch fibers include decreased myoglobin and mitochondrial volume. Both are found to be lower in cheetahs since they require more glycolytic functioning in their muscles. Also, high levels of pyruvate kinase and lactate dehydrogenase are observed in their hind limb muscles. These enzymes catalyze metabolic reactions involved in anaerobic glycolysis. Influenced by increased quantities of fast twitch fibers, this provides evidence of increased efficiency of anaerobic metabolism and limited aerobic ability.
The ability for the cheetah to reach high speeds is essential to successfully catch prey and to ultimately survive. Their musculature allows them to create torque within joints quickly, contributing to faster swing of the limbs and greater stride length and frequency. Large amounts of type 2 fibers produce large amounts of power for rapid acceleration to max speed. By utilizing anaerobic glycolysis, a long recovery period is required after each sprint to return to pre-sprint state.